Hello, everyone. Welcome to Double Canopy. I'm Joe, and today we are talking with Dr. Heidi Jayne Hawkins, who is in Cape Town, and here I am in California. Early morning. Well, not that early anymore. It's almost 7:30 here. She is a research fellow at Conservation International, a research associate at the University of Cape Town, and is the lead author of a new study, Mycorrhizal mycelium as a global carbon pool, which was just published on June 5th. Congratulations, Doctor.
Oh, thanks very much, Joe. And also, thanks very much for the invitation onto your podcast.
Of course.
And please call me Heidi.
Okay.
Yeah.
Doctor sounds so cool. You don't prefer that?
Well, I have to admit, if I'm going to a bank or some other institution where they tend to be impressed by a title, then I use it because it helps. But
Yes.
otherwise, yeah.
Okay. I understand that. Yeah. So Heidi, what has the reception of your paper been like so far?
Well, we were expecting that we would get some attention because in the title we've got fungi, or we are talking about fungi, and we're also talking about carbon. And we know that carbon is a hot topic regarding climate change mitigation. And there's been quite a lot in the news or in popular literature about fungi lately. So we knew it would be quite popular, but we were actually really overwhelmed by the amount of media attention. Um, so there's been, it says, if I look at one of the metrics that I've got here, about 100 news items. I think a lot of those might be just bots, but there's at least 20 genuine news items about our article, and it's been tweeted over 600 times. So yeah, we were blown away.
Mm-hmm.
It's really great.
That's interesting. The first thing you said, the title of the paper, I think correlates perfectly to what the paper's about. It's not like you're writing clickbait, but that's interesting to think that the keywords and the title of a paper can be a draw itself, the headline.
I think so, because there's so much scientific literature, just like other information. There's just so much. And so if you don't get people's attention with your graphics and with your title and your summary, then, people may just move on.
Mm-hmm.
Yeah.
So I think I saw it originally linked to on Twitter by Merlin Sheldrake, who I don't think I was following, but I am following now. And he worked on the paper, obviously.
That's right. And he's well known for his book, Entangled Life.
Mm-hmm. Yeah, and he's got quite a few Twitter followers. I
Yeah.
imagine that is just a huge thing. I mean, getting the word out on social media now. It's just like that's how people are consuming their news and hearing about things and getting information out.
Yeah, that's right. And actually, there's a really nice academic community on Twitter,
Mm-hmm.
and it's quite an easy way to keep up to date with the literature. So if I'm following, a number of people that I'm really interested in their work, and then if I don't see a paper, there's a good chance they'll see a paper and they'll put it on Twitter. And so I find Twitter really useful
Mm-hmm.
uh,
Mm-hmm.
for keeping up to date.
Yeah. I just had an idea and I think somebody does this on YouTube. They'll do a 2-minute summary of the paper. I think we should do that for science papers because you're in your car, you're driving around. I wish I could be reading this right now, but to hear it would
Right.
be terrific.
Right. I think it's a brilliant idea. What they're doing now more and more is graphical abstracts or
Mm-hmm.
summaries. So that helps a lot. And I know that Nature, one of the big journals, is doing what they call immersive summary or something like that, where there'll be some text, but there'll be some video and photos as well. I think it's quite new, but an audio clip, I think that would be really
Mm-hmm.
useful for a lot of people.
Mm-hmm.
Yeah.
Yep. So
Yeah.
I ask everyone because interesting to me. I have teenagers, so I always try to think like what's effective to get them into whatever it is. But what's the effective inspiration? But what inspired you as a young person to become a scientist interested in soil health and ecology?
Well, I think my parents really had a positive influence on me. They always encouraged me to be outdoors, or we did a lot outdoors, and also to be curious about the natural world. Both of my parents are sort of self-taught or autodidacts, I think is the word, where they themselves didn't get the opportunity for a tertiary education, but they are or were such avid readers of science and, well, all sorts of subjects actually.
Mm-hmm.
But I suppose at some point, I spent a lot of time digging around in the garden, and at some point, I developed a fascination for how things work and also things that are quite small or maybe unseen. So, the underground parts of plants and soil. I just developed a fascination for it. It's hard to say why you fall in love with a particular thing.
Yeah.
Yes, I've always loved plants, and the miracle, I think, of photosynthesis is what really grabbed me.
Do you remember how young you were, staring at the dirt and being having questions in your mind?
Well, I do remember being about, well, according to my mother, sort of from 3 years old, digging around. I think most kids like getting a bit dirty, so that's
Mm-hmm.
probably not
Sure.
unusual.
Yeah.
But I spent hours alone in, we were very lucky to have a big garden on one stage and just building things with mud and asking a lot of questions to my mother, apparently. And yeah, I suppose in the middle of um, primary school, I really developed fascination for plants and, this miracle of converting sunlight into energy.
Mm-hmm. So speaking of converting into energy, mycorrhiza fungi is I don't know. I don't know. Do you have a sense of how many people even know about it? Because the thing I find it's soil science isn't really. I mean, it's becoming more mainstream, but it's not. It doesn't seem like it's in the curriculum in the primary schools and.
Well, I think that things like foraging for mushrooms have been in the past, and maybe now there's also a resurgence in foraging for food, including mushrooms. But there's definitely a lot of confusion about what's the difference between a fungus and a mushroom. And I don't think many people have heard of mycorrhizas. So even though I know a group of people who are really quite expert at gathering mushrooms in the forest, I was surprised to hear that actually most of them don't know what a mycorrhizal fungus is.
Mm-hmm.
So no,
And
I don't think it is very well known, although there have been there's been a couple of books now, like we said, Merlin Sheldrake's Entangled Life, and then also a couple of films on Netflix
right.
and other.
Mm-hmm. Yeah, Paul Stamets and Louie
Paul
Schwartzberg.
Stamets
Yeah,
and
fantastic
others.
fungi.
Yeah.
Mm-hmm.
Yes, that's right.
Yeah, I helped fund that movie on Kickstarter.
Oh, really?
Yeah,
Okay.
yeah, they started that on
Yeah.
Kickstarter. Great project. Plants, green plants, soak up and breathe in the carbon dioxide, and they transfer it to mycorrhizal fungi. So you said fungi. Some people say
Yeah.
fungi or fungi.
R-
What's correct?
Right.
What's the
I
right?
don't think there's any
Okay, good.
correct way. Yeah.
I don't
I
want to. I
think
don't
that
want to
the
sound dumb.
I think some British tend to say fungi. I was brought up saying fungi. I don't know what you say in the US, what you tend to say, but
I've heard different
it's
things.
all
Yeah.
it's all fine. It's all
Okay.
fine.
Okay. Good. But the plants feed the fungi. Fungi.
That's
Carbon.
correct. Yeah, fungi, fungi. I say fungi.
We'll get over it.
Um, right. We don't know exactly how this partnership started, but yes, the fungi that live below ground, they're soil fungi, so they don't have green leaves like a plant. They don't have any way of using sunlight to make energy. So they germinate in the soil. There is some signaling that goes on and they grow towards the plant and eventually will join with the root. And then the plant, which is taking up carbon dioxide and converting it into sugars with the help of sunlight and water, then supplies the fungus with carbohydrates, mostly in the form of sugars, at the level of the root. So it's almost like a handshake at at the cellular level
Mm-hmm.
of the root.
Mm-hmm.
And then once the fungus has got some fuel, it can then continue to branch out away from the root. So it's this, and that is what the mycelium is. It is a lot of these branched filaments or hyphae
The hyphae.
called.
Right. Right.
Yeah, but the mycelial network is all those filaments of hyphae that then can spread out around the root, and effectively they're extending the area that's available to the plant root. And so you'll have the roots, but then beyond that, you'll have the fungus spreading out.
Mm-hmm.
And not only that, if you can imagine a root compared to the size of the tiny little threads that you might see on your bread mold, which is also a fungus,
Mm-hmm.
those threads are really small, so they can go into spaces, pore spaces in the soil that the plant can't get to. And that's where the other side of the partnership comes in, because the fungus can access nutrients and
Right.
water, actually, and transport that to the plant.
Right.
And so then you have this exchange taking place at the level of the root. And there are different types of mycorrhizal fungi. Some will actually go right inside the cell of the root, and some will just sit outside the cells. But either
Right.
way, there's an interface there
Right.
where that exchange takes place.
Yeah. Now, the different types, I knew about arbuscular and ecto mycorrhiza, but I didn't know about ericoid or orchid or
Yeah.
the orchid type. And when I was reading your paper, I was just like, oh wow, there's of course it's bigger than I knew, but very complex.
Yeah. Yes. So I think I like, if people are not familiar with mycorrhizal fungi, I like to talk about the mushrooms first and then explain that those are the ectomycorrhizal fungi, because most people would have seen mushrooms growing, and not all mushrooms. So mushrooms are just the fruiting body,
Right.
the above ground part of the below ground fungus. So not every mushroom that you see will be a mycorrhizal or an ectomycorrhizal fungus. You
Mm-hmm.
may have ones that are just rotting wood and are saprobes or saprophytic. But anyway, a good deal of the mushrooms that you see are the fruiting bodies of this type called ectomycorrhizal. The ectomycorrhizal fungi are very common with the conifers and broadleaf trees, including a lot of northern Europe and the northern USA. And they do also form partnerships with a lot of other types of trees and either more herbaceous, softer plants,
Mm-hmm.
but they're really common on the conifers. And then the arbuscular mycorrhiza, they're a lot more cryptic because you don't have an above ground fruiting body that you can say, oh, look, there's a
Right.
arbuscular
Right.
mycorrhizal fungus. Everything happens below ground. So they've got that mycelium, and then they also make spores, but that all happens underground. And they are really ubiquitous. They form partnerships with about 70% of plants on the planet.
Right. And they're each one of those plants, they have they're intracellular. They're going into the cell structure. Is that right? Or on some
So
of
for the
them, yeah, they're arbuscular.
arbuscular, yes. What they do is they actually go through the cell wall of
Oh,
the root. If you've
right.
got the root cell, you've got a cell in plants, you've got the cell wall, then you've got a cell membrane. So they actually go through that cell wall, and then they don't pierce the membrane because that
Okay.
would really
Right.
damage
Right.
the plant. But they form these structures called arbuscules, which means little tree, just because the shape is very often a bit like a hand, like that, uh, or tree-like. So what it does, the fungus will sort of push in that membrane. It doesn't break it though. And then that's your sort of new interface. That's what's happening with the arbuscular mycorrhiza.
And is that what
Where
you've referred to as the Hartig net? Is that
no,
okay?
so with the arbuscular mycorrhiza, the main place where the exchange takes place is this arbuscule, this little sort of tree that pushes the cell membrane of the root cells in. The Hartig net is characteristic of the
Oh,
ectomycorrhiza.
okay. I see.
Yeah, so what happens there is that the fungus will come up to the root and the fungus will go in between the cells of the root.
I see.
So if you've got cells sitting next to each other, you've got cell wall, but there's a little bit of a gap there where fluid and nutrients and things can move. And it's in that little gap that the fungus will form this network, which is called the Hartig net. So it's just a little bit different in that it doesn't go through the cell wall, but it sits right outside.
Right.
And it's the same principle.
I'm curious
And
what level of magnification do we need to see this happening?
so at about if you have a dissecting microscope, so at about times 40,
Okay.
so if you have just taken roots from the field
Mm-hmm.
and you put them into a little dish with a little bit of fluid, and if you have a dissecting microscope, you look at it about times 40. If you've got arbuscular mycorrhizal fungi, you may see the threads coming out
Right.
from around the root. It's not very easy to see because they are often clear. Sometimes they're a little bit colored and you can see. But where you can really start to see something is if you stain them.
Mm-hmm.
So there are stains that will bring up the fungus. And then, so you will be able to see them like that. But if you want to see details like the arbuscule and the spores and the hyphal threads really clearly, then you would want to go up to 100 or 200 or even 400 times. So then you would need a light microscope.
Mm-hmm. And did you guys do any imaging for this study?
So
Video
not
or
for this, well, not for this study, but one of the authors on the paper, Rachel Cargill, she's a student of Toby Kears, who is the senior author. She's the last author on the paper.
Mm-hmm.
And quite independently of this particular study, they've been working on imaging a lot. And you might have seen on the press release, there was a little video of what looked like some strings and then some fluorescent bits moving through.
Yes.
And that was where they had put a fluorescent tag onto some carbohydrates and fed it to the fungus I think they used a sterile culture,
Mm-hmm.
which just makes it a little bit easier to see these things under the microscope, where you haven't got soil and everything floating around.
Everything else.
Yeah, but for this particular study, no, we didn't do any imaging. We gathered data from a whole lot of existing studies, and some of those studies had a lot of really nice imagery where they had, a lot of these experiments that we put together in our study, what they tend to do is supply carbon dioxide to the plant, but that carbon dioxide has been labeled, either a radioactive label, the carbon atom is either radioactive, or it is not necessarily radioactive, but it's still a very different form of the carbon to what is common.
Mm-hmm.
So it's a label that you can then trace. So they would feed it to the plant, they would know how much they've given the plant, and this would usually happen within some kind of confined space where you can control the amount of gas
Mm-hmm.
and the flow and so on. Anyway, and then that would go through the plant, and then that would go to the root, and then that would end up in the fungus. very often they would, if it was a pot experiment, they would have a compartment of the pot where only the fungal hyphae could enter.
Hmm.
And that just gave them a neat way to harvest the fungus only.
Right.
Otherwise it's quite a mixture of roots and fungus, and it's
Right.
difficult to separate them.
Yes.
So then they, at the end of the experiment, they could harvest that all. They could harvest the hyphae, they could harvest the root, they could harvest the shoot of the plant. They knew how much they supplied overall, and then they could say, okay, of the carbon dioxide that was taken up by the plant, so much percent ended up in the fungus.
Mm-hmm.
So a lot of the studies that we used followed that sort of protocol, whether it was in a pot or in the field. And it's not perfect
Sure.
because it doesn't account for the label that might have gone elsewhere, like, there's a whole lot of fungus which is in the root, and that wasn't necessarily accounted for. Or some of the label might have ended up being in the soil after it was given off by the roots or the mycelium. But it's the best approximation that we've been able to make so far. And
Right.
that's what we relied on mostly. Yeah.
Right.
I think one of the fascinating stories is how the symbiosis all got started.
Mm-hmm.
I don't know if that's something
Yeah, I
you asked me about.
I don't know about that. It seems like talking about Paul Stamets again.
Yeah.
I can't remember what it was from, but it was from a little documentary where he was talking about early Earth. Well, not even that early, but about 400 million years ago, um, on Earth, and how fungus and these networks of living things transformed the atmosphere by starting to pull out carbon dioxide and other things out of the atmosphere.
Well, I think what we know is that the carbon dioxide concentration of the atmosphere during the sort of Paleozoic, the time frame that you're talking about 450 million years ago, it was indeed much higher, around 3,000, a little bit over 3,000 parts per million.
It's wild.
But yeah, but the drawdown of carbon dioxide, what we think we know, is really due to plants.
Mm-hmm.
But the spread of plants across the globe, we think, was very much enabled by this symbiosis with the mycorrhizal fungus. So it's like through the plant and through the partnership with the fungus, the plants were able to then quite rapidly, we know that there was a really rapid diversification of the angiosperms, the flowering plants, and the gymnosperms across the whole planet.
Right.
And then that led to the drawdown. So I would say the two things went hand in hand.
Mm-hmm.
And the reason why that happened is because, well, from the fossil record, it looks like those very early land plants, they had really rudimentary root systems. Very much like if you grab a handful of moss now and you turn it over, you'll see there's some thread-like
Mm-hmm.
roots there, but they're not. They function mostly just to hold the plant onto whatever they're on, and that's what the early land plants had. They didn't really have proper root systems, so you can imagine they'd come from an aquatic environment where they were just swimming. Nutrients and water were literally everywhere,
Right.
and now they had to deal with quite a harsh environment of soil, probably quite poor soils at that time,
Yeah, just
because there wasn't.
mostly regolith. Right.
Right,
Yeah.
very very skeletal, not much organic matter,
Mm-hmm.
and now it wouldn't have held a lot of water. Maybe nutrients were really limiting, and so then that's where the fungus would have come in and as we were discussing earlier, would have extended those quite poor roots and brought in water and nutrients, and then, in exchange, get the sugars from the plant, and it would have gone from there.
Mm-hmm.
That's what we think. There's some really nice fossil evidence from a couple of places. One of the well-known ones is in Scotland, where you can actually see these very early fungi colonizing the plant.
Incredible.
Yeah, it is really.
So,
Yeah.
I mean, my mind goes straight to the present. And when we hear people on the world stage talk about climate change and the problems we're facing now, none of them ever bring up mushrooms and this symbiotic relationship, this the ability of plants and mushroom or fungi to draw out, be a carbon sink. But I mean, your paper proves that that's still happening, that it's still possible, and will be possible into the future, far into the future. Why isn't it big
Well,
a part of the larger conversation?
well, exactly. I think that this paper has drawn people's attention to how much carbon dioxide is being drawn below ground by these fungi. And of course, it's been going on for millennia. The problem now is that with the deforestation or degradation of natural ecosystems, that we're losing not only the plants, obviously, and that diversity, but also the soil carbon that is in organic matter, but also in these fungi.
Mm-hmm.
And as we say in the paper, there's some uncertainty about, once the carbon has reached the fungus, how permanent is it? And we're still working on that. But
Mm-hmm.
we think it's, there's large amounts of carbon moving through the system, and that system is functioning in the whole carbon cycle. So it's really important to protect the soil, yes, but also to be aware of this whole symbiosis, because I think that can really affect especially how you restore an area
Mm-hmm.
or how you manage agriculture. I think those are the two big places where
Absolutely.
this information could actually change what you do.
Mm-hmm.
Because, for instance, if an area's been invaded by an alien plant, or there's been forestry the native plant's been cut down and there's been forestry there, you may have completely changed the soil environment and lost the original soil microbes that were there before. So now, if you try to restore that area, yes, maybe they can come in from the side, the adjoining areas, or blow in on the wind. But in some cases, you may have to actually help that along.
Right.
And then you'd need to know, okay, well, we've got these types of plants. What sort of mycorrhizal fungi would be helpful here? Do we
Mm-hmm.
need to inoculate? Sometimes you don't need to. It may be just a case of something as simple as taking some soil from an adjoining area and adding it.
Gotcha.
Uh, and then in agriculture, you would want to encourage conditions that allow these networks to flourish. So that would be disturbing the soil as little as possible.
Right. Right.
And those aren't new techniques. There's no-till,
Right.
there's cover cropping. Also having a diversity of crops, and crops that are perennial rather than being annual,
Mm-hmm.
because with the perennial, as you can imagine, then the connection with the fungus is going to be continuous, at least for some time, whereas with annual, you're harvesting, you're disturbing, breaking up that whole
Right.
network. Yeah, so I think in some ways it gives us new information on how to manage and restore an area, and in other ways it just supports our knowledge about sustainable practices in agriculture that we have already.
Right. Yeah. Aside from the research you've done, do you have a sense of the commercial market for? Because I know there's supplements you can get, inoculants, mycorrhizal fungi. I talked to a guy who sells them, Michael Martin
Yeah.
Melendres, in New Mexico. He sells it. I bought some. I haven't seen a huge difference in my lawn, but maybe.
Oh, I'm so glad you brought this up, actually, because it's not something I know a lot about, except that I know that there are a lot of commercial products out there that have no quality control.
Mm-hmm.
So my colleague on the paper, co-author Katie Field, they actually just put out a paper. I can't remember the exact title, but it's something about we can't rely on mycorrhizal fungi to fix our problems, and that it is more about encouraging conditions that allow them to grow. That was their one point. And
Right.
the other point is that they propose a quality control framework for these commercial inoculants because what they found was that a lot of commercial inoculants sometimes don't even have active
Right.
propagules
Right.
in them, or they have only certain species that might not be appropriate for where you are wanting to inoculate, and then you're
Sure.
just you don't even know what you're introducing there.
Right.
Um, and anyway, there are quality control issues, and they are proposing that we really try and address this now, which I think is a good thing because sometimes you may be encouraged to buy something, whereas and how are you now going to know whether it's made a difference or not without doing some sort of
Right.
controlled trial, you
Right.
know?
Yeah.
Um, it's a bit like, or, buying supplements for yourself.
Mm-hmm.
Sometimes they may indeed be necessary and helpful, and other times it's not really making a difference.
Right. Yeah. So thinking about just soil microorganisms in general, I mean, beyond soil, they're everywhere. They're absolutely
Mm,
ubiquitous, but fragile too. And obviously, the hyphae of mycorrhizal fungi is very fragile. How do these organisms get around? They have to be below ground. I mean, some microorganisms float on the wind and they come in and colonize an area.
yes,
Is that is that the
yes,
same?
yes,
Okay.
yes.
Through
So
spores.
spores,
Mm-hmm.
spores can move in with the wind, with soil. Quite a lot of soil moves around
Yes.
as
No,
dust.
it's huge. It's a huge amount.
It
Soil
is,
transportation.
and right, and then spores will also be in the dung of animals. It's not something I know a lot about, but I'm sure that they move around with animals as well.
Mm-hmm.
It's a good question though. How do Okay, I think with the fruiting bodies of some fungi like mushrooms, that's it's very obvious that those
Right.
get eaten by animals and those get moved around like that. They also get blown with the wind. For the more cryptic ones, for sure they get moved with the wind. I would imagine they must get eaten by microfauna. I
Mm-hmm.
mean, for sure, they are eaten by microfauna, and then those microfauna are eaten by bigger things, and so on, and they get distributed like
Right.
that.
Right. So going back to what your colleague said, creating the right conditions for the things to grow is might be all that we need to do. We might not need to add or inoculate.
Indeed, I would definitely do that first. So that would be, if it's your own garden or if it's an agricultural field, would be to make sure that you have appropriate nutrients for whatever you're trying to grow,
Mm-hmm.
and also to have organic matter in your soil and to mulch your soil. And
Right. Lots of mulch.
yeah.
Never ending mulch. I just wanted to be clear about this, and I think you mentioned it later in your paper, but your study shows with just the ectomycorrhiza that about 13% of the carbon pulled in from the plant
Hmm.
goes to the fungi in exchange for whatever the mycorrhiza is giving back to the plant. It just seems very significant.
It's huge. And if you actually look at those plots that we have in the paper, I think what's even more interesting is the variation. So for both the arbuscular and the ectomycorrhizal fungi, you get instances where up to 50% of the plant's photosynthetic carbon is being allocated to the fungus. So that's a huge amount.
That is huge. Is that energy mostly going to hyphae growth and spreading
Yeah,
out?
Yes,
Okay.
that's right.
And the other question I had was with all these different species of fungi, are they all symbiotic or do they compete with each other, like the ecto and the arbuscular mycorrhiza? Are they in competition?
So there are studies on that, but there are really not enough studies where you've got mixed communities of these different types of mycorrhizal fungi, even not enough studies with mixed ectomycorrhizal fungi, because for sure in nature you will not just have one,
Right.
and the same with the arbusculus. So we don't have enough information on that. However, there is clear data that some of the saprophytic fungi in the soil, so those fungi that are just living independent of plants and breaking
Oh.
down bits of wood and
Mm-hmm.
organic matter,
Yep.
they will compete with an ectomycorrhizal fungus. So there's some really cool studies where they show you can see into the pot and you can see how the ectomycorrhizal fungus spreads out away from the plant in the soil. And when you have a saprophytic fungus there, then the extent to which it spreads out is much reduced. So that fungus, that saprophytic fungus, is sort of creating is coming in one direction, and the ectomycorrhiza is coming in another direction, and there's sort of a front there, and no doubt all sorts of
Mm-hmm.
interesting chemistry going on.
Mm-hmm.
But certainly, microorganisms do compete with one another, and we know that a plant having arbuscular mycorrhizal fungus will be an advantage in that it is less vulnerable to disease, including diseases caused by other fungi.
Okay, so
You know,
that's really interesting because you said that, or your paper says that arbuscular mycorrhiza doesn't live as long as the ecto or other types. So what happens to the living plant when you have these intracellular networks that die?
well, it's an ongoing process. So what we meant by that is that particular structures inside the plant have a certain lifetime. The arbuscules will be there for some time, and then they will disappear, they will dissolve. But there are other parts of the same fungus that are colonizing the plant, or there are new micro arbuscular fungi that are entering the plant. So it's not that there's a gap. It's just that a particular structure or part of the fungus will be relatively shorter lived than, say, a lot of the ectomycorrhizal.
Okay.
And then that dead fungal matter will also persist in the soil, at least for some time. We know that the fungal hyphae
Necromass.
necromass is a great word. I like it.
I was reading it
Yeah,
and I was like, this would be a good name for like a metal band. Fungal necromass.
yeah, right. It's a great word. Anyway, the living and the dead hyphae, contributing to the soil organic carbon. And not only that, they have benefits for the soil structure itself because they are themselves thread-like, and so they bind soil particles together, but they also exude sticky compounds that
Mm-hmm.
make soil aggregates, and that sort of protects the organic carbon, and it also means that you've got a better water holding capacity in that
Right.
soil, less susceptible to erosion and
Right.
like that. Well, I've gone off on a bit of a
No,
tangent from
it's terrific. Thank you. I'll ask you one more question. I like to hear
okay.
people's if they have a perspective or have noticed anything in popular media. Obviously, there's a big one right now, but movies or TV shows related to your field or what you study. Or do you have any favorites?
What was that one you mentioned by
Oh, Fantastic Fungi. Yeah,
fantastic fungi?
documentary.
I'm trying to remember the names of others.
It could be a
Well,
monster movie. It doesn't have to be a documentary. Is there?
right.
There's I mean, right now the popular one on HBO is The Last of Us, which deals with fungi. Have you heard of
I
that?
haven't. I've heard of it, but I haven't seen it.
Okay.
So I've read Merlin Sheldrake's Entangled Life. I think it's brilliant
Hmm.
because it really popularized the underground and these networks, and besides mycorrhizal fungi, just the whole ecosystem that's underground. So I think
Yeah.
that is my favorite. I really like the movies as well because, for the same reason, it is brought the underground into people's awareness. I do find that there tends to be a mixture of fact and a little bit of stretching of the fact and even some misinformation in those movies. I think that that's probably inevitable,
Hmm.
and it's just the scientist's job to keep trying to be involved in these sorts of documentaries or films
Right.
and, try and get it as close to what we think we know as possible.
Sure. Um, I've had very limited experience with press, but what I have had is has been it just seems like it's really difficult to have a lot of information, compress it, and then have it be coherent or fact based
Accurate.
on the other end of it. Have you noticed anything in the press coverage for your paper? Or, has it been mostly correct or broadly true or not?
I think it's been broadly true. I think with this paper of ours, what a lot of people have understandably picked up on is that number we gave of 30%, 36% of our emissions from fossil fuel, that the 13 gigatons of carbon dioxide equivalents equates to about 36% of our emissions from fossil fuels. That is, it's staggering. It's a
It's
mind-blowing
per year.
amount
Per year.
per year. Per year, that's right.
That
Thanks.
yeah, that's incredible.
And then we do go on to say we know this is how much on average is coming into the system. We don't know how permanent it is because of course it's a flux, right? It's
Hmm.
coming in. Some of it is definitely leaving as carbon dioxide again out of the soil. Some of it is becoming this wonderful necromass and even binding onto soil particles. So we need to get that right. I think we've tried to keep the wording clear. So we've said plants allocate carbon, 13 gigatons, about carbon dioxide equivalents per year to the fungus, instead of saying, oh, the fungus sequesters this amount or the fungus stores this amount, because those words aren't really right, because storing implies that it's staying there,
Right.
and sequester actually implies technically that it's an amount that is increasing,
Right.
ever more increasing over time, and we don't know that yet. So that's why we use the word allocate. A lot of news platforms will sort of say, well, what is allocated? Sort of an awkward word, and then
Yeah.
they'll just use another word.
Right.
But
Well, if you'll permit me to bring one other thing up, I was reading just this morning about Australia's soil carbon credit system that they've developed over the past 10 years, and it's a strange system. I don't know how they're going to be able to do it sustainably, especially if more people do it. It just seems like a really big project, but it relies on measuring carbon in the soil through
Mhm.
various methods. And I mean, that's essentially what you did. You measured the biomass of mycorrhiza.
Right, so we use studies that
Right.
calculated the numbers, and in many cases they would have harvested the biomass. Yes.
Right. So to do that in an incentive scheme, they would need to do that. I mean, in perpetuate forever.
Hmm.
They would need to keep doing
Yes.
that over, every year.
Well, when people take soil samples, they will be in these carbon offset projects. They will be wanting to measure soil organic carbon, and they'll take that whole soil sample, they'll send it off for analysis, and what they get back will be a combination of carbon from plant litter remains, soil organisms, soil microorganisms, including mycorrhiza. So there's no need in those carbon offset projects for them to say, oh, this is how much mycorrhiza there is, or where that carbon actually came from. I think all they want to demonstrate, and all that is really feasible to demonstrate, is that whatever their changed practice is, whether it's no-till agriculture or it's less grazing, that they're showing that intervention is having some effect on the soil organic carbon. Now, where it gets tricky is that soil organic carbon tends to increase only very slowly. So you may have an intervention, but it might take you 10, 20, 30 years to
Yeah,
show
their first
something in the soil.
their first interval is 5 years, and then
Resample.
you have to retain those numbers, at least those numbers for 25 years,
Mm.
or you have to give the credit back.
Right. That's where modeling comes in.
Mm-hmm.
So it understandably, it's impossible to show that you're going to have an effect without measuring it, but you don't want to wait 30 years to get your carbon credit. So then you have to develop a model based on actual measurements that you make at the beginning of the project or just before the project. And then based on that, using models, you will predict with your intervention how much carbon you're going to sequester. Now, I think that is one area where we could improve, because some of those models are based on they're good, and they're real workhorses of these types of projects, but they are based on outdated knowledge. So there are people working on including mycorrhiza and other processes that we know are happening into these models. So updating these models and making them more realistic. Because I think now, decades down the road, we are seeing that a lot of these carbon offset projects are failing because they're not delivering on what they said they could.
Right.
And sometimes it's because they're just poorly done, but sometimes it's because these models perhaps are over predicting. Or if you take a soil sample here, and then you try and come back in 5 years and take it in the same place, but you haven't taken it in exactly the same place. Soil is inherently heterogeneous, and then you have to just take a lot of samples. So, I mean, some carbon offset projects that rely on plants, so like forest reforestation, are a little bit simpler to manage. But the ones that really rely a lot on soil carbon, I think they are worthwhile as long as they are done in an ecologically and socially appropriate way, definitely, but they are challenging.
Well, Dr. Hawkins, I really appreciate your time. It's been a pleasure to talk with you, and I'm really inspired by your work and hope to see it flourish and, more studies like it, and hope you continue to do it as well. Thank you so much.
For sure. Thanks very much. Thanks for taking me.
Of course.